Can you guys introduce yourselves?
I'm Nicole Zaragoza.
My name is Carla Rodriguez.
Well, I'm from Sabana Grande, Puerto Rico, and I have been in Franco's lab for about a year and a half.
I'm from Vega Baja, Puerto Rico, and I have been with the Franco Lab as a postdoctoral fellow for about a year and 2 months.
I think it all goes back to my great-grandmother's house. I love her. I was raised with her. She had a lot of plants, and so we spent a lot of time in her garden, and I would play with like snails. And I always wondered, how do they go into the shell? And it was just, curiosity.
I guess as a kid, I grew up in the beach a lot, living in Puerto Rico. And I was always fascinated by, I always wanted to be a mermaid. So I spent most of my time in water, snorkeling. So at first I actually wanted to be a marine biologist, just so that I could be in the water all the time. And then later on, that transformed more into, if you liked science, all that I saw was that you have to be a doctor, because I'd never really had any exposure to research. And then I took this really awesome course, molecular biology of the cell, and I really became interested in how cells work, because they're so complicated and everything happens at a certain time and it is specific, and it works so perfectly, and we're all here just by the individual work of cells. That to me is just completely fascinating.
Personally, it was because Puerto Rican populations face very unique healthcare issues and are usually underrepresented in a lot of clinical studies. So I think just being able to really understand the genetics of our local population can help us figure out new ways in which patients could benefit. Maybe the therapies that are currently in place are not necessarily the best for our population. So how do we understand our patients better so that we can improve their quality of life as well.
I feel really lucky that I was able to find Dr. Franco's lab and do my postdoc here. It's been an amazing experience. I feel like the research that we do here, somehow it has more impact than when I felt more like just a cog in a machine over there, but here I feel like we're driving the research, and we're trying to be more impactful and trying to recruit, like Nicole said, we're trying to really build a pipeline to get tumors from patients here and study tumors from patients here. We just published a paper on these two proteins that are important in the involvement of breast cancer. And so we just published that, and following up on that same project, my newer project now is truly my baby. I feel like, because it's more driven towards what I want to do, which is find new therapeutic avenues. So we found this really interesting protein that might be important for therapy resistance in breast cancer. And so what my hope is that we're able to show that if you target this drug in this population of cells, then you could treat these patients that are no longer responding to current therapies. And so right now we barely just started, but that's my goal for the future, to be able to take it all the way. That would really be my dream. Yeah.
Currently working on single cell sequencing techniques. These are tools that allow us to really look at changes in the gene expression of cancers at the individual cell level. You can really dive into what are the specific populations or the specific cell states that are driving this cancer, and to better understand them. Something that I'm excited for is the fact that we're trying to create this pipeline to really facilitate the process of getting patients and recruiting them to get the tumors and have more samples so that can be more representative of our community. It's new technology here, and it's really important for us to really get to explore our community and really understand cancer here.